Hello, and thank you for joining us today for BioTek's webinar event. My name is Lindsay, and I will be the moderator for today's webinar. The audio for today's webinar will be coming from your computer speakers, so please be sure they are turned on and the volume is up. To ask a question, please use the chat feature. It is located in the bottom left-hand corner of your screen. You can send in your questions at any time. Our speakers will address the questions during the Q&A session at the conclusion of the event. If we do not get to your question, we will do our best to respond by email. Today's webinar will be presented by Dr. James Hynes, Head of Research and Development at Luxcel Biosciences, and Wendy Goodrich, Application Scientist at BioTek Instruments. We'd like to thank them for joining us today. With that, I'll turn things over to James to get us started.
Hi Lindsay, and welcome to everybody. Thank you for the introduction. As Lindsay mentioned, my name is James Hynes. I am Head of R&D with Luxcel Biosciences. And I suppose, first of all, to take a brief look at what we do as a company, we are involved in the development specifically of oxygen and pH sensors and the development of systems and assays around those sensors across three main areas. And those are food and industrial packaging testing, rapid microbiology testing, and the focus for today, which is cell biology and life science research tools. So in the area of food and industrial packaging, with our US partners MOCON, we have developed the first handheld oxygen sensing system. It's an optical system, and we've developed the oxygen sensors, which allow the measurement of gaseous oxygen in a contactless manner. The underlying technology is also common to a technology platform called Greenlight, which allows the measurement of bacterial growth using screw cap vials with an integrated oxygen sensor. And they're used across a number of different areas, from in-process testing to process development to food safety applications. And finally, for the focus for today is looking at cell biology life science research tools. So this is a suite of products which we use to essentially measure metabolism on plate reader platforms. And we'll go through today how those technologies work and how they can be applied to the assessment of metabolism. So to take a very quick look at, I suppose, an outline for my short section today, I thought we'd start with maybe a little refresher on metabolism for people that haven't thought about this since they were undergraduates. So it's a very simple look at metabolism, just to place in context the measurements that we'll be speaking about later in the presentation. From there, we'll take a look at a means by which cellular oxygen consumption can be measured using a product called MitoXpress Extra. Then on to how we can look at glycolysis on a plate reader using a product called pH Extra, and also how these two products can be combined to give a more holistic overview of how the cell is responding to a particular treatment using drug screening as an example. We'll also take a look at how one might measure cellular oxygenation and maybe consider why that's important and how all three parameters can be particularly relevant using an area of aerobic glycolysis or Warburg as an example. So that's a very quick overview of the content of the talk. So to begin with Metabolism 101, as I'll call it, so this might be very, very simple for some listeners or maybe a useful refresher for others. But to start with the mitochondria itself, and it's often viewed as this sort of bean-shaped organelle whose sole function is to produce ATP, purely because of, I suppose, the outdated diagrams that people often see in textbooks. Now, the ATP generating part of the mitochondria is obviously hugely important. When you consider that your average male turns over his body weight in ATP each day, then an impairment to that system obviously causes significant problems. But over the last 10 to 15 years, it's become increasingly apparent that the mitochondria has not just got one sole function. It's critically integrated into the processes of the cell, and it's more accurate to consider it as a reticulum rather than an individual organelle. So it's a highly dynamic reticulum, changing all of the time and heavily integrated into the cellular signaling pathways, particularly with organelles such as the smooth endoplasmic reticulum. And because of that, it's been implicated in a variety of disease states and a variety of signaling mechanisms. So interest in this particular area has grown considerably because of that over the last maybe 10 to 15 years. Going back to, I suppose, a very simple overview of metabolism, and I've just said that, it's not just simply a bean-shaped organelle, so please excuse my drawing, because here we're looking at bean-shaped organelles. But to take a very simplistic view of the cell, here we've got a cartoon diagram of the cell. We've got the cell membrane here. We've got the mitochondria here. And we're looking at what happens as metabolism in this cell proceeds. Looking mainly at glucose-grown cells, because a lot of the cells used in vitro models are typically grown on glucose. But through the presentation, we'll also consider other substrates because they can be critically important in ensuring that the in vitro models used for these type of metabolic testing are as relevant to the in vivo condition as possible. So moving to what happens as glucose enters the cell. If you consider that glucose enters the cell, enters glycolysis, with glycolysis producing pyruvate and ATP. That's, I suppose, the first step of what we're considering. That pyruvate has sort of got two routes that it can take. And again, this is a simplistic analysis, but that pyruvate can either be converted to lactate, which is exported from the cell, or the acid that derives from it is exported from the cell, causing extracellular acidification. Or it can enter the TCA cycle, or Krebs cycle, which in turn generates CO2 and generates reducing equivalents for oxidative phosphorylation. Now, OXPHOS is essentially the activity of the electron transport chain, which generates mitochondrial membrane potential, and that membrane potential then drives a whole variety of cellular processes, including the generation of ATP. So ATP is produced because of the passage of electrons along the electron transport chain. Those electrons, as we've said, derive from NADH or FADH2, depending on the source, and they finish up with molecular oxygen. So the process that we're measuring when we assess oxygen consumption through the rest of the talk, we're essentially measuring the activity of the electron transport chain, which in turn is giving us information on OXPHOS, which is one of the main sources of cellular ATP. Now, there are other substrates which are important, as we've mentioned at the start, and one which is particularly important is glutamine, something that's been maybe overlooked for quite a long period of time, but it's becoming increasingly apparent that it plays a critical role in a variety of cellular metabolisms and can be a particularly important source for the generation of ATP. Obviously, beta oxidation and other sources of reducing equivalents are also important, but just to keep things simple for today, we'll focus on glucose. So if we take a look at what we can measure, and we spoke about three products at the start of the talk, the first one to consider is the measurement of glycolysis. In an open system, the vast majority of the acidification that you will see in your in vitro system is because of the conversion of pyruvate to lactate. So if we measure the rate at which the cell is acidifying the media that it's in, that gives us some idea of how hard glycolysis is running. We can also measure oxygen consumption, the rate at which cells are depleting the oxygen in the environment that they're in, and that gives us some very useful and important information on how hard the electron transport chain is running, really how hard OXPHOS is running, which is the second main ATP generating pathway. So if we take a look at those two measurements, what we're doing is, in a convenient manner on a plate reader, we are generating information on the two main ATP generating pathways within the cell. Now finally today what we'll take a look at is how one can assess the level of oxygen available to the cell. Because obviously the cell will perform differently or will react differently in conditions where there is lots of oxygen available in comparison to conditions where there is very small amounts of oxygen available. So that parameter is critically important and often overlooked. So we'll take a look at how those measurements can be made and the impact that those levels can have on cellular metabolism. So moving from there to the technology that we're going to use to do those measurements, as we've mentioned, there are three. One will assess oxygen consumption, the second will assess extracellular acidification and by extension glycolytic flux, and the third will look at intracellular oxygen. Now to bundle all of these three probes together and to consider them as one, I suppose, system, they all perform in a very similar way. The oxygen probe particularly, the more oxygen you have, essentially, the less signal you have. And if we start in an in vitro assay, and we start at, we'll call them normoxic conditions. Now that's an argument we could have as to what normoxia actually is. But if you start an in vitro assay, you typically start at 21% oxygen. As cells or isolated mitochondria or enzymes consume the oxygen in that well of the microtiter plate, that causes a reduction in oxygen concentration. That reduction in oxygen concentration causes an increase in signal, and that increase in signal is what we're measuring. Now, whether we're measuring oxygen consumption or oxygenation, the process by the underpinning, the technology that underpins that is the same, and it's a very, very similar system when we're measuring extracellular acidification. So to take a look in a little bit more detail at the spectra of the types of probes we're talking about, purely to look at how we could maybe use them to measure other relevant parameters, this is the excitation, or a typical excitation spectrum for the types of dyes and probes that we use, and this is an example of an emission spectrum from that same technology. Now, you'll see that there is a significant signal change when you deoxygenate the sample. So we're going from here a normalized value of 1 to here a normalized value of 5, so that's a 5-fold increase in signal. So the probes respond very strongly to the reduction in oxygen in the well, which provides good performance when we're measuring these parameters. The second parameter to take a look at is where we excite the probes and where we measure the probes. So we typically excite at about 380 nanometers, and we measure emission at about 650 nanometers. Now, anybody who does work with fluorescent probes or works with fluorescent assays will realize that that's quite a big span. And what that does for us, particularly on plate reader platforms, is that it allows us to multiplex with other metabolically relevant endpoints, such as mitochondrial membrane potential and other measures of metabolism that are important. We can do that either on the same plate or in a lot of instances in the same well, because of the spectral flexibility of the measurements that we're doing, and because of this big gap that we've got between where we excite the technology and where we measure the emission. So to go from there and to take a look at what is happening in the well and the protocols that we use to do these measurements. So if we start with a typical 96 well plate, this type of technology can be applied to basically anything that consumes oxygen. We apply it to bacteria, to yeast, to enzymes, to small aquatic organisms, to tissue homogenates. But for today's talk, just in the interest of time, I'm going to focus on the eukaryotic applications for in vitro testing, so basically isolated mitochondria and whole cells. So here we've got cells at the bottom of the well. We resuspend our probe in growth media, and it will work with whatever media you're using. You add that media with the probe in it to your 96 well plate. We then overlay with a layer of mineral oil, and that layer of mineral oil is there to prevent oxygen, or to at least slow down considerably oxygen diffusing from the environment back into the sample. What happens then is the cells themselves consume oxygen, as we've seen earlier in the presentation, through the activity of the electron transport chain, and that causes the concentration of oxygen in the well to reduce. So we go from atmospheric levels of oxygen to much lower levels of oxygen. That causes a signal increase. So we go from low levels of signal to high levels of signal. So to recap, the cells are consuming the oxygen reduces in concentration in the well, and that causes an increase in signal. And the type of readout that delivers, we measure these conditions over time. If we have a condition where there's no change in oxygen concentration in the well, we see no signal change. If we have a condition where oxygen is depleting in the well, this would be typically our untreated condition, we see the signal increasing. If we're comparing that to a condition where we're seeing reduced metabolic activity, reduced mitochondrial activity, we see that as a lower rate of signal increase. And that would be this instance here. If we have a condition where oxygen consumption has increased, such as the addition of an uncoupler or the activation of uncoupling proteins, then we see that as a faster rate of signal change, indicative of the electron transport chain basically working much harder. So that's a very simple overview of how the technology works. It's important to realize that it's a very flexible system. It can be done on 384 well plates or 96 well plates, or all sorts of other sort of bespoke systems. In any instance where we've got an oxygen change happening in the sample, the probe will pick that up. But from an assay setup point of view, it's important that we have as many cells in the well as is relevant to the application, so that the signal changes can be as strong as possible. So moving from there and applying that system to analyzing isolated mitochondria in the first instance, and then some examples of measuring cellular respiration. As an example of isolated mitochondria, we're just going to take a look at the measurement of a rat liver mitochondria in this instance, and we're going from a high concentration of mitochondria to a low concentration of mitochondria, and you'll see that the replicates are very tight, and we're seeing concentration-dependent signal increases. And this type of system is used for, a wide variety of applications. One of the commonest would be the assessment of the mitochondrial toxicity of particular drug compounds. This is an example of a dose response with a classical inhibitor. This is KCN, an inhibitor of complex 4. And you'll see that as we move from high concentrations to low concentrations, the rate of signal increase is getting slower. So here we've got untreated samples, and we've got a strong rate of signal change. Here we've got higher concentrations, and we've got no signal change. So that facilitates high throughput assessment of the effect of drug compounds on the activity of the mitochondria, and it also allows dose response information to be generated quite quickly in the sense that you can assess these across a 96 or a 384 well plate. For anybody who works with mitochondria, it also offers the possibility of measuring state 2 and state 3 in parallel, so with and without ADP, and also measuring your full panel of electron transport chain substrates, again, all on the same plate. So instead of with some traditional systems whereby you've got to wait and do these measurements sequentially, with isolated mitochondria, that can be problematic because, activity can drop off over time. The fact that you can measure them in parallel means that you can get a lot of information from a single mitochondrial prep. As I've mentioned, this particular example is using rat liver mitochondria. So when one is screening individual compounds, you've got a plate map that you will kind of view the data on any of the plate readers that would be doing the measurements, and Wendy will go into this in much more detail as to how the instrumentation is outputting those systems and how they're optimized on the BioTek measurements. But this is just an example of 2 wells measured in duplicate, and that's identifying an inhibitor. And you can see on the plate there are lots of other inhibitors flagging. And this is specifically looking for inhibitors because we're looking in the presence of ADP. So that's an example of how MitoXpress Extra can be used to assess mitochondrial function, and by extension how it can be used to screen for the mitochondrial liabilities of a panel of compounds. Now moving from there to measuring whole cells, obviously the technology can be applied to whatever cell type you're working with. We've worked with a wide variety of cells, and recently we're doing quite a lot of work with stem cell derived hepatocytes and cardiomyocytes, primary cells, humanized cells, because of the drive, I suppose, towards a more in vivo-like cell type, with people trying to improve the biological relevance of the measurements that they're doing. This specific example is using stem cell derived hepatocytes from Cellular. On the left we've got metabolism happening. On the right we've got inhibited cells, so no signal change. So you'll see a very strong Z factor for anybody who's interested in the performance of these types of assays. The Zs on these cells particularly are up about 6.9 to 7, so that's good performance. What we can do then is we can look at the effect of compounds on the metabolism of the whole cell rather than just the isolated organelle. And if we take a look at a panel of electron transport chain modulators, we can start with oligomycin, which blocks the production of ATP via the ATPase. That causes immediate inhibition of oxygen consumption because of the fact that you're not allowing mitochondrial membrane potential to dissipate. We can then take a look at 2 electron transport chain inhibitors, rotenone, which inhibits complex 1, and antimycin, which inhibits complex 3, and both of those cause an immediate reduction in oxygen concentration. We can also measure uncoupling, and you get mechanistic information from this. Again, this is a classical uncoupler where we treat with FCCP, and that causes an increase in oxygen consumption because of the dissipation of mitochondrial membrane potential that it causes. So that's just using the classical electron transport chain modulators to demonstrate how the assay can be used to assess the mitochondrial function of a panel of cells. We do a lot of the work in 2D systems, and the users do a lot of work in 2D systems, but more and more people are turning to various 3D models. And the system is quite applicable to 3D models because this particular probe is measuring changes in the media that the cells are in. So there are no issues with lifting cells off 3D matrices, which can prove problematic for some other read types. So whether you're measuring on collagen matrices such as the RAPT system or others, or some other hydrogels or matrigels, or whether you're measuring on polystyrene scaffolds as we have here with the BioTek system, or some electrospun scaffolds as we have here, the same measurement type is possible. You're measuring on the same microtiter plate platform, but you're measuring in 3D, which for some applications is a particular advantage. So moving from there to, I suppose, maybe a halfway house between the isolated organelle and the whole cell. Measuring the whole cell is obviously has got considerable advantages, but in some instances, people will want to deliver either compounds directly to the mitochondria or substrates directly to the individual complexes of the electron transport chain without having to isolate mitochondria from the cells. Now, one means by which that can be done is to permeabilize the cells with something like digitonin. There are a menu of options, but digitonin would be one common example. So essentially what you do, without going into the mechanism that this happens, but a simplistic view of it is basically that you punch holes in the cell membrane. And because the cell membrane is subtly different from the mitochondrial membrane remains intact. So by punching holes in the membrane, you can study the mitochondrial function mechanistically by allowing electrons access to individual complexes of the electron transport chain. In this instance, the first thing you would need to do is to optimize what concentration of digitonin is needed, because different cells require different concentrations. And just, I suppose, to show how the system works and to show how optimization can be done, here we're just showing the effect of increasing digitonin concentrations in the presence of rotenone and succinate. Now, what we're doing is the succinate is impermeable to the cell membrane, so it cannot pass through the cell membrane unless we've successfully punched a hole in the membrane. And we've got the cells sitting in rotenone, which means that succinate is essentially the only substrate that can be used. So other reducing equivalents from glucose can't enter through complex 1. So we are blocking this complex, and we are allowing substrate through this complex. And only when we see respiration do we have permeabilization. So that allows us to say that in this instance, for this cell type, the appropriate permeabilization concentration for digitonin is here. And that's also, I suppose, demonstrating that we are now delivering reducing equivalents to the individual complexes, which was not previously possible. If we were doing this mechanistically, what this would tell us is that the compound we had added, if we didn't know that it was rotenone, was blocking complex 1, because when we add to complex 2, we are now seeing respiration. So that type of mechanistic information can be generated, and there are a menu of options as to how you deliver electrons to complex 3 and onto complex 4 for those types of analyses to identify the specific site of the insult if one has already identified that the compound is causing a problem. So that's mechanistic analysis using cell permeabilization, in this instance using digitonin to achieve that. So that's MitoXpress Extra and how it can be used to assess mitochondrial function in whole cells and in isolated organelles. As we mentioned at the start, it can also be important to get some sort of a picture as to what the cell is doing from a glycolytic point of view. And to do that, we use pH Extra. Now, pH Extra is a pH-sensitive fluorophore that we add to the wells in the same way as we do MitoXpress Extra. And to take a very quick look at specifically what we're measuring, again, our simple, overly simplified view of metabolism, we have glucose entering the cell, entering glycolysis, in one instance generating lactate, which eventually through a number of steps causes the export of hydrogen ions, or entering the TCA cycle for oxphos. When we measure acidification in an open system, we are mainly measuring hydrogen ions deriving from lactate production. When we measure in a sealed system, we're also measuring hydrogen ions that derive from the production of CO2. Now, because in most instances for these types of applications, glycolysis is really the thing that we're interested in, then we measure in an open system. But done. Now, what that allows us to do is to measure extracellular acidification on a 96-well plate on the type of instrumentation that Wendy is going to describe later. So simply what we do is we degas the plate to remove CO2 from the plastic so that we are sure that the acidification that we measure is because of biologically derived hydrogen ions and not just CO2 leaking from the plastic back into the sample. You resuspend a buffer tablet that arrives with the kit to give you your respiration buffer that is simply sterile filtered and pH'd. It's added to the wells with your pH extra reagent, added to the plate, and that's read. Now the type of data that you generate looks a little like this. So here we've got a high concentration of cells causing rapid signal change, which indicates rapid pH change, which indicates rapid acidification. And as we said, in an open system, that is typically due to the production of lactate. As we drop the cell concentration, as you would expect, the rate of acidification falls. So that's just showing how the number of cells in the well affects the rate of acidification. Now, I suppose what's a little bit more relevant to the parameter that we're talking about is what happens when we stop the cell from processing glucose through glycolysis. So if we use 2-deoxyglucose to block the hexokinase, we would expect to see a significant and dose-dependent decrease in acidification rate, and you can see that's exactly what we see. So as we increase the concentration of 2-deoxyglucose from 0.1 to 10 millimolar in this instance, you see a significant, reproducible, and dose-dependent decrease in acidification. Now, this type of assay can be used obviously to assess glycolysis or glucose metabolism in and of itself, whether you're looking at glucose transporters or you're looking at particular modulators of glycolytic metabolism. In the interest of time, what I've done is I've just looked at some of the applications, or an example of an application whereby we look at both this parameter and the parameter that we've just seen. So if we take a situation where we are looking at both oxygen consumption, so the activity of OXPHOS, and extracellular acidification, the activity of glycolysis, as we said, we're looking at the two main ATP generating pathways. And that allows us to get behind some of the mechanisms by which cells can circumvent mitochondrial insult. And I suppose to emphasize that point, or to underscore the value of these dual measurements, if you take cells that are exposed to a panel of electron transport chain modulators, these are the same ones we've seen earlier in the presentation. Rotenone inhibits complex 1, antimycin inhibits complex 3, and FCCP is an uncoupler. Despite the fact that we know these are potent mitochondrial poisons, if you look at cellular ATP in the vast majority of cell lines, 1 hour post-treatment and up to much further post-treatment, you see little, if any, change in ATP. So here we're seeing no change in ATP post 1 hour. That's despite the fact that we know these compounds considerably affect oxygen consumption. So now we're looking at oxygen consumption. These are the red bars. We've got complete shutdown with rotenone, complete shutdown with antimycin, and uncoupling with FCCP. And the question is, what's actually happening there? And this underscored, I suppose, a weakness with the cell models that for a long time, albeit changing more recently, were used to assess mitochondrial toxicity within, I suppose, the pharma and biotech industries. What the cell is doing is it is upregulating or increasing flux through glycolysis to maintain ATP in that manner. So here you see glycolytic flux increasing significantly in response to the mitochondrial insults that we've caused through the addition of rotenone or antimycin. And really what we're looking at is the balance between the flux heading in this direction and the flux heading in this direction. So most cells in the body will be driving most of the pyruvate in this direction. Some cancer cell types will drive a lot of it in this direction, but we'll discuss that later. But a lot of cells in culture, typically those sitting at high concentrations of glucose for extended periods of time, have the capacity to switch from one to the other. So if we block OXPHOS and we block the ability of the cell to generate ATP aerobically, it simply increases flux through glycolysis and generates ATP anaerobically. So by measuring both parameters at the same time, we have a more complete picture of how the cell is responding to that particular treatment. So I suppose what that underscores is that standard cell models coupled with standard cell viability assays are particularly insensitive to mitochondrial dysfunction for that reason. Measurement of oxygen consumption significantly detects or sensitively detects mitochondrial dysfunction. And when both probes are used together, we have a very simple but powerful way of assessing the flux through the two main ATP generating pathways. Now, as mentioned earlier, when you look at the spectra, we can multiplex with other parameters also, such as JC1, which will give us some information on mitochondrial membrane potential, and some ROS probes, which will give us an idea of how much ROS is being produced, to give, a multi-parametric assessment of metabolic perturbation from the particular treatment. That can be used for screening, as we've said. If we take compounds which cause an increase in acidification and an increase in oxygen or a decrease in oxygen consumption, then they are typically mitochondrial inhibitors. So these are mitochondrial inhibitors in this quadrant of this type of analysis. So a decrease in oxygen consumption and an increase in extracellular acidification. We can also identify uncouplers because they cause an increase in acidification and an increase in oxygen consumption. So you can do this type of mechanistic analysis by, from a drug screening point of view by measuring both parameters in parallel. It also allows us to profile cell types. So different cell types have different metabolic poise. Some are more dependent on extracellular acidification, some are more dependent on oxygen consumption. This is data generated by Dr. Carol Morton in Oxford. And you can see that cells kind of at this part of the spectrum are more dependent on oxygen consumption, less dependent on acidification. Cells down here are much more dependent on extracellular acidification, so are more glycolytic and rather than oxidative. And it also allows, I suppose, an analysis of the importance of the substrates that are used. So if we take a look here, we've got oxygen consumption and extracellular acidification for a particular cell type at increasing or decreasing glucose concentrations. Typically, cells are measured at 20 millimolar glucose, but you can see as these cells are exposed to lower concentrations of glucose, you have a significant shift in how they're metabolizing. They're becoming more and more dependent on OXPHOS as we drop the glucose concentration. And that opens the question, maybe for another day, as to what the relevant glucose concentrations are. 25 millimolar is a huge concentration of glucose, despite the fact that it's very commonly used. Or also, what is the relevant substrate at all? Because a lot of cell types would not really preferentially use glucose in the body. So moving from that to the assessment of cellular oxygen consumption and aerobic glycolysis, we've spoken about using 3D maybe to increase the biological relevance of cell in vitro cell models. People use that. We've just taken a look at, what is the relevant glucose concentration, and maybe beyond that, what's the relevant substrate? Should we be using glutamine? Should we be using fatty acids? But an often overlooked parameter when assessing, the biological relevance of an in vitro model is the oxygen concentration that should be used for the analysis. So very, very briefly as an example, to take a look at, first of all, how oxygenation can be measured conveniently on a plate reader, and secondly, how it's related to this concept of aerobic glycolysis or the Warburg effect. To make the point, I suppose, first of all, that despite the fact that a lot of people culture cells at 21% oxygen, most tissues in the body never see, in fact all tissues in the body never see 21% oxygen, and the tissues that people typically study would be normally experiencing a much lower oxygen concentration in the body. Now that will depend on the organ that they're in, the part of the organ that they're in, but if we take a look in really rough terms at the kidney, the liver, and maybe even the brain, we're going from oxygen concentrations from 10 down to maybe 5%. Significantly lower than the 21% that are typically measured. Now that significantly affects the metabolism of the cell, and it also can affect cell signaling because of the involvement of oxygen in particular cell signaling pathways. But the fundamental question is, how can this actually be measured? And what we're taking a look at here again is the balance between these two parameters and how the concentration of oxygen can affect that balance. So if we look at aerobic glycolysis or the Warburg effect, again, it's got a long and interesting history, but I suppose as it's currently understood, it is essentially the conversion of glucose to lactate, or the running of glucose or of glycolysis even in the presence of oxygen. But obviously, knowledge of how much oxygen is actually present is key to an accurate understanding of the models being studied. So what we're looking at is what is this balance here and what is that actual concentration. So the question is, I suppose, what is the relevant applied oxygen concentration? Can it be monitored in 2 and 3D models? And how does that oxygen concentration affect flux? So the last slide just looks at that. So here we've got an intracellular oxygen probe. It's a nanoparticulate probe that is loaded into the cells. And here we're imaging that probe on the citation 3, and Wendy will look at that in more detail. But as we drop the oxygen concentration, we see an increase in signal, as we've discussed from the start of the presentation. Now this, at the moment, is not a quantitative measurement. However, if we do the measurement on the plate reader, as you would conventionally do, using a dual read that, again, Wendy will describe in a bit more detail, then we have the capacity to generate quantitative information on the oxygen concentrations at the base of your well, either in 2D systems or in 3D systems. And what these measurements show us is that there is a significant disparity between the oxygen concentration that is applied and the oxygen concentration that the cells actually experience. So this purple line is the oxygen concentration that the cells are being exposed to in the unit. And this instrument allows the oxygen concentration to be modulated over time. But if we measure what's happening in the cells, we see a significant difference between what is set on the unit and what the cells are experiencing, because these cells are consuming a lot of that available oxygen. And those deltas can be anything up to 10%, depending on the oxygen concentrations and depending on the cell densities and depending on how much metabolism is actually happening. So obviously, the more oxygen being consumed by the cells, then the less oxygen available to the cells, essentially. Here we've got cells sitting at 10%. If we decrease their oxygen consumption rate through the addition of an inhibitor, then they return almost to atmospheric because they're no longer consuming oxygen. If we increase their metabolism, then they go to almost complete anoxia. We're down at about 2%, despite the fact that these cells would be sitting in culture at about 14%. So depending on how hard they're working, the actual oxygen concentration that the cells are exposed to can be significantly lower. And to finish on the last point, the question then is, how does that affect the balance between aerobic metabolism and anaerobic metabolism? And if we take a simple condition of 6% oxygen and 21% oxygen, you find an increased dependence on glycolytic metabolism and increased extracellular acidification at these lower oxygen concentrations for the very reason that the cell is denied oxygen or is being supplied with less oxygen and is now more dependent on extracellular or on glycolysis as a source of ATP. So to summarize, MitoXpress Extra, we measure oxygen consumption. It's a key indicator of mitochondrial function. pH Extra is a measure in an open system of glycolytic flux. Parallel measurements allow us to assess the balance between aerobic metabolism or aerobic ATP generation and glycolytic ATP generation, and we can multiplex with a variety of parameters. We can apply to mitochondrial tox screening, and that allows us to sensitively detect mitochondrial toxicity. Those screens can be done in isolated mitochondria or in whole cells, including 3D systems. And finally, we can apply to a variety of disease states. We're just picking, I suppose, maybe a very crude interrogation of cancer metabolism and the Warburg effect on those last few slides. But the general point is that we can assess the interplay between available oxygen, aerobic ATP generation, and glycolytic ATP generation. All of those can be done on the same plate, and they can be multiplexed with other parameters which give other useful information on metabolism. ATP can be measured on the same cells after any of these measurements, while other parameters such as mitochondrial membrane potential or ROS can be measured kinetically at the same time using the instrumentation that Wendy will now describe. So with that, I'll hand over to Wendy for a discussion of how the measurements we've just looked at can be performed on the BioTek instrumentation.
Thank you, James. So Through our initial collaboration with Luxcel, we worked to optimize and validate the probe detection, all 3 probes, and the data reduction principles on our readers and the imager. We have an application note, a poster, and sample files on our websites to assist any customers in running the probes on a BioTek reader. I'm going to highlight some of the validation work we did here, but I also want to emphasize that probe signals, cell densities, cell lines, or other detection and imaging parameters be optimized by each individual end user. So James really covered the detection principle pretty thoroughly, so I'm just going to highlight it here quickly, that the higher the signal from the probe, the lower the oxygen and the higher the pH. These are the 3 MitoXpress probes that James explained. I'm going to spend just 1 minute talking about the multiplexing, since James really did highlight that a bit in his speech. And although we don't show the data here, we did do a proof of principle on the multiplexing, not necessarily with other parameters, but with the probes themselves. And one of the first things we optimized were the filter ranges so that we could use the same excitation filters for both the intra and extracellular probe and the pH probe. Those probes do have their own emission filters, however, that I'll talk about in a little bit. But to run all 3 probes on the instrument, you only need the 1 excitation filter. And we loaded the plate with the probes both individually in wells, and then we also combined them into some of the wells to multiplex them. And then we did 2 separate read steps, one at the first excitation and emission wavelength and one at the second. And there was a really good assay window between the individual probes, and then when they were both in the same well together, multiplexed, those individual read steps distinguish those probes as well. So that's just shows some of the flexibility for running these probes in the microplate. They're also, the probes have 3 detection modes. I think the pH probe now is really optimized for TRF, but the other probes can also be detected in RFU, standard TRF, and then the lifetime measurement. And some of the data you'll be seeing is in different detection modes, but we found that the lifetime measurement was the most sensitive and had the higher assay windows. It also allowed direct conversion of lifetime to pH calibration, H+ quantification, and extracellular acidification rate. Also, you can go directly to oxygen consumption rates. Our Gen 5 software can integrate each of these detection steps and convert the data into lifetime, and I'll show you that a little later. So James again also went over the excitation and emission spectra here. There are only 2 things I want to point out that he didn't mention. The first I mentioned on the first slide, which is that we optimized the probes so that they both, the excitation 380/20 filter can be used for all 3 probes, and then here we have the emission wavelengths here. We optimized at 645/15, and this one was at 610. So that shows us here. The other thing I want to point out here is in the lifetime measurement. That's actually an equation that takes the ratio here under the curve of the fluorescent probe, the decay of the probe, and you can measure at these 2 different time-resolved steps. And we did everything at 30/70, and this is explained more in the application note. But you can also edit these. They can be 40/60. You can try 20/80, and either way, you may be able to gain some sensitivity on the assay. So the instruments that we optimized for the probes include our Citation 3 imaging reader, our Neo 2 reader, the Synergy 2, and the H1. The main difference between these, especially with the Synergy 2, this does not have gas control. So if you really want to control your CO2 or your O2 environment, the Synergy 2 is not compatible for that. These other readers are. There's a few other accessories you might want to think about when doing these assays. All of these systems come with dual injectors so that you can add compounds at different times during your read. And those can be switched out so that although you may be able to start with 2 injectors, you can, without interrupting your read, swap those out and put another set in if you want to continue to add more than 2 compounds to your plate over the kinetic interval. The other thing I want to point out here is that for imaging, although we've only done initial sort of proof of principle work on imaging these probes, we now have a laser cube, which really reduces photobleaching and phototoxicity effects, which for this assay, especially because it's done over long kinetic intervals and repeated detection and imaging steps, the probes can be subject especially to photobleaching. So this laser cube is really a great addition for the Luxcel probe imaging. We also have a new BioSpa, and this is a self-contained incubator and CO2 chamber. You can control both CO2 and O2 at the same time within the chambers, and this moves plates between the incubator and a dispenser or washer and a reader, and it can house, as you see, up to 8 different plates or vessels of any kind. This really can help allow hypoxia culture to be conducted on a small footprint. Plates can be rapidly transferred to an O2 controlled reader for investigation. There's a delidding option on the robot, so you can put lids off and on in the plate. So it's a nice option for environments that don't have a enclosed hypoxia workstation or dedicated low O2 incubators. So I just wanted to highlight that. So I was going to show a video here, but I think we're running a little low on time, so I'm going to skip that for today. But I would point you to our website, www.biotek.com, and this video on the BioSpa 8 is right on our homepage. And you can just click on it. It's about 2.5 minutes long, and it's a lively, entertaining look at how the BioSpa 8 can improve your life in the lab. So the application note, this is a really good place to start if you're going to be using these probes on a BioTek instrument. We've done a lot of the initial work and have these recommended settings for depending on the reader and the probe that you're using. It's a very good place to start. This is also in the application note, which is available on the website. And it just shows some of the different parameters that we looked at during optimization. One of the things I want to point out is that when you're gaining on these probes in the detection mode, we did both gaining on a high well and a low well, and since these probes increase or decrease according to oxygen levels or pH levels in the plate, it may be useful to start by gaining on a low well where there's very little probe detection rather than on a high well, because as oxygen decreases or if hypoxia conditions increase, then your probe signal will get much, much brighter. We do now offer an extended dynamic range on the reader, so if probe signal goes up considerably higher than what you've gained on, you'll still be able to capture detection for that. But these are all things you may want to consider when you're optimizing the probes on your reader. And here are some of the parameters. Again, this is a list. I'm not going to run through it, but here are some of the parameters you may want to consider when you start to create your workflow for your analysis or investigation. Things like your filters, top or bottom read, your lifetime window, your plate type and map. That would be for full plate and partial plate because it's a kinetic assay and we have sort of a custom algorithm for the lifetime detection. Reading a full plate or a partial plate can have some time difference in getting your final detection number. You may want to look at different oils besides a sensitive oil. There's a high sensitivity oil that comes in one of the kits, and you may want to use a different oil, or you may want to look at the difference between a top and bottom read. If you have the oil overlay on the top, you may want to always read from the bottom, although you'll see in some data I'm about to show that it doesn't really make that much of a difference from a detection point of view. You'll also want to try different cell lines and cell densities, probe concentrations, times and temperatures, etc. So here is what you can expect for using different emission filter band passes. And what this really shows is that the narrower your band pass, the lower your background and the greater your sensitivity will be. So for example, if you're considering using the 360/40 excitation filter for the pH measurement as an example, you may get better performance, and we did get better performance when we switched it to the 380/20. And so all three probes could use the same excitation filter. And this shows us here, this blue line here. Can we see? No. Yeah. This here is the narrower band pass. And this here is a narrow band pass, so you see that the incident light is much lower from a lower blank, and the transmitted light is higher. So that's what you're looking for when you're optimizing your filters. We also did a performance comparison on the top and bottom reads. As I said before, this may affect if you have an oil overlay, and the take-home message here is that the top optics have less variability and the bottom optics provide a wider assay window. We did partial and full plate reads again because the lifetime measurement is two separate read steps at different time resolved parameter settings. We wanted to make sure that the data between reading just part of a plate or a full plate was the same, and you can see here that it's highly correlative, so it doesn't really matter how many wells on your plate you're running, the lifetime measurement is still fully compatible. We did cell density optimization here. We did untreated and then the uncoupler and the inhibitor, and this is just different cell densities. That one of the things is because some of the investigations might happen over a longer kinetic interval, you may want more cells to track over the investigative time. And then we just took a 60K, which is the green here, and we applied the compound here to get oxygen consumption inhibition. So this is just showing you some of the things. The one note I want to point out, I'm sorry, I stumbled there, was that the pH extra and the extracellular probe both require one overnight incubation, but the intracellular probe requires two. So you may want to adjust your cell density numbers based on that as well, and also that cell line dependent. Some cells grow very, very quickly and some grow slower. So when you're optimizing your cell densities, you might want to consider that as well. So we then applied the optimization here. This is a nice demonstration of signal conversion, the lifetime signal conversion to extracellular acidification rates. We used a pre-calibrated value here to do these conversions. This is also in the application notes, so if you need more detail on that. But the kit inserts also provide a very easy, simple to use pH calibration method if you want to fully optimize this calibration number here. So we have here is the lifetime measurement from the reader, and here's the pH scale. Then this is the conversion to H+, and the conversion to extracellular acidification rate. And this was the delta pH here. We just showed that it was within about 1.5 to 1.6% of the original pH. Then on the right here, these graphs just really show the MOA of the pH extra probe, where as acidification increases or pH decreases, the probes increase. I'm going to just fly through this stuff here. Here's intracellular O2 levels in response to drug treatment. We have uncoupling here, and then we have inhibition here. This is antimyosin. This is different concentrations of that, and this is dose response to the different concentrations. And this is for the intracellular O2. This was done on the NEO. And again, this is also shown in the application note. I'm going to go quickly here. We're running out of time. Here's the extracellular oxygen consumption rate, also in response to drug treatment. You can see that the inhibition here is low and the uncoupling is high. We get a nice assay window here, and we did the data on this linear portion here. And then the O2 stepping, we manually adjusted the O2 levels inside the citation 3, and we did the lifetime measurements and imaging at the same time over the 3-hour period. This just sort of shows again the probe signal increase as oxygen decreases. Here's the stepping here for the compound treatments. And finally, we're working on a proof of principle here, multiplex dye imaging. We did a DAPI stain on the nucleus here. This intracellular probe, the Luxcel intracellular probe, is in the cytosol of the cell, and this is the nuclear stain, so we then overlaid them here. You can do things here like mask to do cell counting, which is, other parameters, so you can normalize the cell number over the kinetic run. And we also have looked at, although we didn't show it, we looked at the 3D scaffolding with the BioTek plates, and initial proof of principle was successful with those as well. And here I also just wanted to note that you can convert your field of view. Here, this is a 20x magnification. You can convert it to a full number of cells in the full well. So you can take the cell count in this field of view and actually convert it into how many cells per well. So here's the signal optimization workflow that we recommend everyone go through. The conclusions really are that the probes can be detected in all 3 detection modes, RFU, standard TRF, and the lifetime. You can get instrumentation that extends the workflow for higher throughput or in highly controlled dedicated oxygen control environments with robotic incubation station, automated dispensers and washers for full cell culture capability and drug treatment analysis. Again, I would point you to the application note on the website. We also have sample files on the website that actually have a lot of these algorithms built right into them and have real data from the readers. And you can also start there. They have oxygen consumption rate, conversions, they have the lifetime conversion, and there are a few other equations in those sample files as well that may help you as you start your optimization. Thank you.
Thanks, Wendy. As you said, we're tight for time, but I might just take a very quick run through some of the questions that have come in, and I'll group them together. Apologies to some people that we don't get to, but as Lindsay said, we'll try and get back to as many people as we can by email. One of the questions was, can neurons be used? The answer is yes. There's some particularly interesting work being done using aligned electrospun fibers on 3D models for neurons particularly. That's applicable. There was a question on how the mitochondrial concentrations shown earlier relate to the actual amount of mitochondria in the well. The simplest way to answer that, I suppose, is to say that our assay volumes are typically about 100 to 150 microliters, so essentially 10 times less than the mg per mil values that you'll receive on your slides. There was a question on whether or not oxygen transfer through the plate affects sensitivity. And yes, it does. I was trying to make that point, but maybe not clear enough on that slide. So what we're measuring is oxygen depletion happening in the well, and we need a level of oxygen consumption that exceeds the level of back diffusion through the plastic. So that's why we would typically recommend using fully confluent monolayers to maximize the response from the cell. So the probe will detect any change in oxygen that happens, but the consumption must exceed back diffusion for that change to happen. I suppose another question was how it differs from seahorse. So without going into it in a huge amount of detail, really what you're looking at are two discrete measurement approaches. One is a dedicated instrument for measuring oxygen consumption and extracellular acidification. The other is a system which is designed to be compatible with plate readers, as we've just seen. So the flexibility that comes with that would be the big difference, in the sense that we can measure other parameters alongside, and we can use whatever type of plates you're typically using. But we can't do the injections, particularly on the samples that are overlaid with oil. Now, injections can be made through the oil, and as Wendy said, injections can be made for the extracellular acidification and intracellular oxygen measurements, but users typically don't do injections for the oxygen consumption measurements. I think that's to answer that one. And a final question was, can the measurements be done at 5% oxygen? And how sensitive is the system at 5%? The answer is, the lower the oxygen concentration, actually performance goes up. And as you've seen, Wendy has used the citation 3 at lower oxygen concentrations. So the answer to that question is a short yes. So I think that summarizes most of the questions that have come in, but as we're tight for time, we're just not able to get to them all. So with that, I'll hand back to Lindsay to wrap up and maybe let you know how you can get some more information on the topics that we've discussed today.
Yeah, I'm just going to answer this last question here. It says the arrow of pH is going up. You mean the acidification. This is the pH here. These levels are going down, which means acidification is increasing, and the probe signal is going up in parallel to that. So this is the signal, and this is the pH.
Perfect.
Great. Well, thank you very much for joining us today, and thank you to James and Wendy for their presentations and for taking time to answer some questions. We will email this recorded event and a transcript to all participants. If you have further questions for our speakers or would like more information on BioTek's products, please visit our website. In addition, please check out our blog, like us on Facebook, and follow us on Twitter to hear more about BioTek webinars or to learn about our products and how they can help you. Please join us at any of our upcoming events. You can view our complete schedule online at biotek.com/events. Again, thank you for joining us.